Okay, is this thing on? If everybody could just file in, we still have some benches in the back, seats in the front. Thank you everybody for coming. It's great to see everyone. See a lot of old friends, which is great. Some new people as well. I'm very appreciative of everyone's interest, and I'm so glad to see everybody tonight. For those of you who don't know, tonight is the third of 4 lectures we're giving here in Southampton by Stony Brook University School of Marine and Atmospheric Sciences. So there's one more after tonight, and that's on May 13th. We have Greg Metzger, who's from Southampton High School, a teacher there, also a Stony Brook graduate student, I believe, officially. But he runs an incredible program on sharks and catching sharks. He works with Chris Paparo. He's got some amazing videos. And this summer, he's going to be running some very interesting programs, tagging sharks with an international organization whose name escapes me right now. But if you have any interest in sharks, I highly suggest you come and see. And it's all local. It's not like he's going to the Caribbean. This is all work that he does in Shinnecock Bay and just south of Shinnecock Bay. And I think you'll be amazed at the diversity of sharks and the incredible videos he has. So that's Friday, May 13th at 7:30. So I hope you can make it. And with that, I'm going to start my lecture. I am tonight's speaker. And this is my 2016 State of the Bays address. And you can see the title for this year is Challenges, Victories, and Solutions. And so, appropriately, well, first I'll acknowledge, hopefully you all came, I know most of you were here early. I hope you all got to see the wonderful posters that are out there put together by the graduate students. I always say they're the real boots on the ground and the fins in the water. You know, my days of being out on boats, collecting samples, and even being in the lab running those samples, those days have passed. And so really everything you're going to see is the very hard, earnest, and studious work of the people working in my lab group. They're shown here. You saw them presenting posters. And so actually, let's give them a hand because they're really the ones who make it all come together. So my outline is essentially the overview of my presentation is the title. And so I'm going to talk about what's happening in the bays, the state of the bays today. And when we talk about victories and solutions, victories maybe is a look back, and solutions is a bit of a look forward. And so I'm going to start with this slide here. And, you know, I think as I prepared this presentation, what I realized is I feel that all of Long Island has gone through a great epiphany in recent years of a few different things. And so one of the great epiphanies, I think, is just shown right here, that we live on an island, it's a watershed, and any water that hits Long Island eventually goes into our aquifer and becomes our drinking water. And then eventually that water seeps out and goes into our coastal waters. And in both cases has a very strong effect. A stat that really was struck me was that fact that people, oftentimes when people think of their drinking water, they may think or say, oh, well, yeah, I don't want to contaminate the drinking water, but I have city water. You know, of course, there's no city water. It comes from Suffolk County. But even people in Suffolk County, if you ask the people from Suffolk County Water Authority, the mean distance between where your drinking water comes from and your tap is 3 miles. Right? And so what that means is that anything you're doing in your neighborhood or your neighbors are doing is affecting your drinking water. And then the epiphany further is the fact that it's also now affecting our coastal waters as well. The other big epiphany in the last few years, I believe, is the recognition that the concentrations of nitrogen in our groundwater here in Suffolk County just continue to rise. And you know, now in hindsight, we recognize why. We know that most of this is due to on-site wastewater. And the fact that we've designed individual homes to have individual septic tanks that are supposed to leach their contents into our groundwater. And that those systems do a very good job of making sure pathogens don't get in our groundwater. They also do a very good job of delivering nitrogen right to our groundwater. And we now know, I'm not going to show another slide on this, so I'm just going to say it, we now know that more than 2/3 of the nitrogen that's responsible for this increase is from on-site wastewater. That is our individual septic tanks and cesspools. And the recognition that this increase is now having profound impacts on our coastal ecosystems. And so to look back at 2015, here's a map that I would put together in collaboration with the Nature Conservancy, showing you everything that happened in 2015 with regards to water quality impairment. And these are impairments, not that I'm just making up, but these are impairments at the federal or state level, violating what New York State DEC wants to see, or what NOAA wants to see, or what US EPA wants to see. And so we have so many things to point out here, so many areas of low oxygen and algal blooms. I can't give a 1-hour lecture to go through all this. I can only give you some of the highlights, or lowlights as it is. But you'll see the issues with regards to low oxygen conditions and algal blooms are widespread. There's really not a region. We at the North Fork, the South Fork, the East End, Western Nassau County, the North Shore, the South Shore, all being afflicted by different types of algal blooms or experiencing low oxygen. And so just to highlight some of these, if we did it chronologically and looked at 2015, there's the very sad tale that I'll tell more about. The season last year, remember it was so cold in the spring last year, and as soon as it warmed up, things started going wrong in our bays. A massive turtle die-off in the Riverhead area, followed by this massive fish kill that occurred in the Peconic River, Flanders Bay, and out to the Peconic Estuary. A brown tide that went all the way from the Nassau County line to here in Southampton that went June into July. In Nassau County, massive seaweed blooms washing up on ocean beaches. And then going into the fall, a rust tide that occurred both in the Peconic Estuary, also in Shinnecock Bay, for the first time ever in western Shinnecock Bay. And so I won't talk about all the details, but what I'll say is that we now know that these higher levels of nitrogen are leading to either harmful algal blooms being more intense, growing faster, or getting more toxic. And you can see all these studies. I'm adding studies year by year. These are international peer-reviewed publications that are fully in line with what's known across the globe about these events. So I'm going to start by talking about one of the most concerning of these events, Alexandrium, which causes PSP. It makes this compound saxitoxin that can be filter-fed into, or filter-feeding bivalves can accumulate that and the toxins, which is a serious human health issue. This is a neurotoxin. It blocks sodium channels. At high levels, it can lead to death. People died in the last few years in Alaska due to this. And below that, it can be paralysis. So it's a serious issue. And what I've done for you is, I don't know how many chemists we have in the room, but if you look at the molecule saxitoxin, what you'll see is 7 atoms of nitrogen. And so the international research on saxitoxin is now very clear. When this organism has more nitrogen, it makes more of this toxin. And when it has less, it makes less. Teresa Hattenrath-Lehman is here, or Dr. Hattenrath-Lehman proved this about 6 years ago, and now there's been an international flood of research showing the same thing. She proved it for right here on Long Island. And this also goes for some other toxins as well. The ones that are nitrogen-rich, if the organism has more nitrogen, they're going to be more toxic. And so, thanks to Teresa's hard work over the last decade or so, we now have a very good sense of where this organism can be found all across Long Island. Anywhere you see a red dot is an area that is more than 1,000 cells per liter, which can make shellfish toxic. And in fact, we had multiple situations of shellfish bed closures last year due to PSP and saxitoxin. So, for example, 2,000 acres of Shinnecock Bay was closed for more than a month last spring because saxitoxin turning up in shellfish. And the epicenter of this was in Weesuck Creek right here in East Quogue, where the shellfish had more than 3 times the federal limit of saxitoxin. In addition, in the Riverhead area, Meetinghouse Creek and Terry Creek were closed. Those are closed to regular shellfishing, but was also closed to gastropods because of their ability to accumulate the toxins. And then a new area was found. This is on the North Fork. This is James Creek, which feeds in the Great Peconic Bay. This is a region, we've been studying this for 10 years. We just happened to be collaborating with the county. We just happened to get some samples, and Teresa saw high levels of cells. She said to the DEC, you may want to check the shellfish. And they had 5 times the federal limit of PSP toxins, meaning that someone could have gotten very sick had they been eating shellfish out of here. So despite the fact we've got a lot of information, there's still surprises out there, unfortunately. And as I mentioned before, Teresa's research has shown not only does more nitrogen make them more toxic, it makes these organisms grow faster. And again, she did this first research published in 2010. There's probably been dozens of publications following up on that showing the same thing globally since that time. Now, I want to hone in on the turtle incident that occurred last spring. And specifically, you can see the headline here about dozens of turtles. And I see Mark Lusty is in the audience there. Mark, wave your hand there so everybody can see who you are. Here's a video showing that he went out on one day last May, I believe it's May 15th. This is very long, but essentially to show you that in a single stroll of one peninsula in Flanders Bay, he counted more than 100 diamondback terrapin turtles. We know that there were more than a half a dozen areas where they washed up. So this was hundreds of diamondback terrapin turtles. A team has been put together to look into what the cause of death was here. Originally, the DEC responded and brought in Cornell University to do necropsies on these turtles. Teresa then worked with the Riverhead Foundation to do some further necropsies. We brought in Big O Laboratories in Maine to do toxin analyses, and we brought in a biologist from Hofstra University to look at the ecology of these organisms. So that's the one peninsula where the turtles were found, but we also know they were found in other locations as well. And just some quick points about diamondback terrapin turtles. You can read all the details here, but the bottom line is they're not very good at reproducing lots of additional diamondback terrapin turtles in a short period of time. They live a long time, but their rate of reproduction is very, very slow. And that's why in neighboring states, well, that's why they're considered a near-threatened species. They're an endangered species in Rhode Island, a threatened species in Massachusetts, species of concern in Delaware and other states along the East Coast and Gulf Coast. Now, looking back at what happened, I told you Meetinghouse Creek was closed due to a PSP. Here's the time course of the bloom that we saw there. It started showing up in late April. Peak of the bloom was by early May. The area was closed by early May, and the level of toxins in the shellfish there was very, very high. So that is nearly 7 times the federal closure limit, and that's a very high dose. The question is, what does that mean for a turtle? Now, before this all began, I'll be perfectly honest with you. I'm not a turtle biologist. When I first heard the idea that, oh, a turtle was poisoned by saxitoxin, I said, well, that's ridiculous. Turtles eat algae, right? Well, that's not true. Diamondback terrapins eat shellfish. So I said, okay, well, I guess it's possible, but, you know, they would eat a lot of mussels, right, because turtles are fairly big. Well, that's also not true. Diamondback terrapin turtles are small. A male weighs ½ pound. A female weighs 1.5 pounds. And so Teresa put together this table. We don't know what a lethal dose is for turtles. Thankfully, no one's going around trying to poison turtles with saxitoxin. But we do have some data on vertebrates and other similar sorts of organisms that we tried to back calculate. She came up with a range that maybe they would need between 10 and 100 micrograms per kilogram of toxin. At the peak toxicity of the shellfish in Meetinghouse Creek, that would mean they would only need to eat a single mussel to get a lethal dose. Even at the higher level, the male would only need about 1 mussel, and the female would need 3. Now, we don't know exactly when everything happened. You know, this is not, it's like CSI, I guess. I've never seen the show, but I guess it's about solving crime stories, right? This is CSI Riverhead. And so we actually did some toxin analysis. This is color-coded here. The different bars correspond to different regions. So the toxin analysis came out like this. Some of the turtles had no toxin whatsoever. Some had levels well beyond what would have been the lethal dose. In hindsight, in talking to the DEC who collected the turtles, what we found is the ones that came out with nothing were ones that had been sitting around for a long time and rotting on the beach. And the ones, for example, that we collected, Mark, who went through all those dead turtles, took the freshest one we could find. And that one, a lethal dose within the heart. And you can see the other ones that were early on and collected right away, also high levels. The other piece of evidence, we also found toxins in the fresher turtles within their guts. We also did, or Teresa, I should say, did DNA analysis. And I have a saying that the DNA doesn't lie. So she did high-throughput sequencing of a gene specific for animals in the guts of the turtles, which would pick up anything they were eating. And sure enough, every turtle had the DNA signature of mussels. And so a clear evidence that each of them were definitely eating mussels. There were other things in there as well, fish. But the study's still ongoing, but what I'll simply say is that all of the scientists involved are in agreement that saxitoxin was the cause of death of these turtles, which is there's not a lot of precedence for, but something to be concerned about going forward. And again, to make the link to nutrient loading, I told you this organism gets more intense and more toxic when it's given more nitrogen, and so that's the link then ultimately to the turtles. Unfortunately, that event was followed very shortly thereafter by a massive fish kill. In fact, the fish kills in the Peconic River area actually started at the same time as the turtle kills. The first ones were smaller scale, didn't catch people's eye. The one that happened at the end of May, however, was hundreds of thousands of fish. The fish spread all throughout the Peconics. You can see this is washing up on the beaches of Flanders Bay. Here's within the Peconic River, estimate of 400,000 fish. There was an official report. You can get this report, and you can read exactly what the conclusions were. I collaborated in this report along with the DEC as well as Suffolk County. And so the official causes of death, and I'm going to show some more detail on this. Firstly, the oxygen conditions were very, very low. You're going to see actually it wasn't low. There was no oxygen in the river at the time of the deaths. We also found, surprisingly, that not only was there no oxygen, but the fish had damage to their gills, so they were not able to breathe. Their breathing was compromised. And then finally, there was a larger than normal number of fish in the Peconic River at the time. So to go through some data, I'll just put it all up and we can talk about it. You can see the time frame. This is towards the end of May into the beginning of June. You can see what happened here. This is levels of nitrate. There was a large thunderstorm event that occurred just before this. We saw a drop in salinity as well. You can see that in the report. The nitrate levels went up. And concurrently, we had an algal bloom that got more and more intense in the river following this input of nitrate. And as the bloom got more intense, here are the oxygen levels. And so what you see is that the bloom, as it's getting towards its peak, the oxygen's getting lower and lower, and then it's going to zero for an extended period of time, 6 to 8 hours. And so if that's not bad enough for the fish, there was this particular dinoflagellate in the river at the time. In fact, there were 3 separate fish kills, and all 3 of them. And when the DEC did some histopathology of the gills, this is what they looked like. Now, I should have had a control, but these are supposed to be nice and straight, not touching each other, because this is the lamellae of the gills where oxygen exchange occurs. These are fused together, and you can see the mucus from the algae accumulating within their gills as well. So, again, people who were from Cornell as well, who did the autopsies on this fish, looked at the gills, concluded that they had compromised gill function due to this algal bloom and the accumulation within their gills. That's being another reason contributing to the fish kill. And as if zero oxygen for 6 to 8 hours wasn't bad enough, they also had damaged gills. Okay, the last of the algal blooms I want to mention move into freshwater. And these are also a very significant issue for human health. There are multiple types of freshwater cyanobacteria that make multiple classes of toxins. If you've heard me talk before, you may have heard the fact that these names, microcystin and anatoxin, came in the late 20th century. In the mid-20th century, when they first discovered these compounds, they were called fast death factor and very fast death factor. And that's true, actually. This is a gastrointestinal toxin. This is a neurotoxin. And these are serious issues beyond even for humans. Here's a study in China that linked gastrointestinal cancer to people who are using surface waters contaminated with microcystis as their source of drinking water. Thankfully, we don't need to rely on that, but we do need to worry about our pets. And so the CDC has confirmed that there's been hundreds of cases of dogs being poisoned by blue-green algae and their toxins. And in fact, if you're curious, this summer, wait till, say, July or August, September, go in Google News and put in blue-green algae and dog, and unfortunately you'll find something about either a dog getting very sick or dying. And you may know that in Georgia Pond, we had a dog die in 2012 from drinking the water, an area that we know has very dense blue-green algae blooms. And last year, at Fort Pond in East Hampton, 2 dogs got sick. They didn't die, thankfully, but got sick following drinking water with a blue-green algae bloom. So this is a serious issue, probably more for our pets than it is for us. Now, thankfully, the DEC is tracking these events across New York State, and for good reason. We use upstate, people are using their surface waters for fresh water, the Great Lakes, for example. And, you know, New York is a great state of many lakes. You can see the Finger Lakes, the Great Lakes, and all sorts of little lakes in between. And there's 62 counties across all of New York State. And can we guess which one had the most blue-green algal blooms in 2016? That would be Suffolk County. Far and away, the largest number. Now, I'm going to be let's be perfectly honest about the data. This is partly a function of occurrence and monitoring. If there's not a monitoring program, they're not going to pick it up. So we're looking here, and we're finding it here in Suffolk County. But I will note that there are 45 counties that have these blooms. So at least in 45 New York State counties, people are looking. And here we have 16 different systems that came up with blue-green algal blooms above the New York State threshold in 2015. And you can see on down from there. Beyond the number of systems, Suffolk County also had the most number of total weeks listed. And again, this is data that's publicly available. So if you want to see it, go to the New York State DEC website. You can download it. They have an annual report they put in. So the most number of weeks. And then here are the top 10 lakes for number of weeks listed in New York State. And Suffolk County with 5 of those lakes. You can see Lake Agawam, Marituca Lake on the North Fork, Lake Agawam's in Southampton, Mill Pond in Watermill, and then Wainscott Pond in East Hampton. Interestingly, my laboratory was designated by New York DEC to be monitoring New York City lakes and ponds. So this past year and the coming year we'll be getting samples from there. And you can see there are some doozies in New York City, specifically Prospect Park and also Central Park Lake as well. Came up many times, and in some cases very intense blooms. And here's year by year the number of lakes and weeks that these events are being reported. And again, part of this is looking for it, but again, we also know that there are 48 other counties that are out and looking. And we do know these events are driven by nutrients. In fact, probably a stronger case here than any of the other algal blooms. So this is an old figure, but if you look at it, log TP is the log of the total amount of phosphorus. As the phosphorus levels increase in a lake, you go from having all sorts of different types of algae to exclusive blue-green algae. But you'll note this is a fairly old figure. This goes back to the 90s. Work we've done since then have shown, this is showing Lake Agawam, that blue-green algae can also respond very strongly to nitrogen. And so when you see the green here, this is microcystis, one of those blue-green algae. In this case, in Lake Agawam, it could care less if you're giving it more P, phosphorus. It's responding to nitrogen, and when you add both, you get almost the same response as nitrogen. So some of these blue-green algae have the ability to what's called fix nitrogen, make their own nitrogen, and that's why people always think phosphorus is important. But here on Long Island, we're getting mostly microcystis, which cannot fix nitrogen, and therefore it is dependent on external nitrogen sources. And that's why the EPA very recently, just last year, has come up with the idea that if you want to prevent eutrophication in coastal ecosystems, specifically lakes, you need to think about both nitrogen and phosphorus. And again, that's in contrast in the past where for lakes, people were only thinking about phosphorus. Okay. Want to move on, talking about last year, talk about the water quality index that our lab has run as a collaboration with News 12, monitoring 30 sites across Long Island. And those 30 sites being reported out, usually by Bill Corbell, and the Thursday night weather forecast between 5:30 and 11:30 at night. And so for this, we're gathering lots of data, and we've accumulated lots of data. And we're learning a lot about the variability of our coastal ecosystems and what makes them in good and not so good shape. I'm going to skip this and just go to some of the data that we're finding. These are the total levels of algae, so total phytoplankton in different systems, all 30 systems. You can't see it all at once, but I'm going to highlight some of the problem areas. Total algae, you'll see most of the areas here are on the South Shore, and you see some areas out by us, Quantuck Bay, Moneybog Bay, Penniman Cove, coming up with some of the highest levels of total algae all across Long Island. Similar with regards to water clarity. Here are the areas from the best to the worst water clarity, and again, you look at the list here, these are some of the same exact places. Some of the worst water clarity all across Long Island. If I didn't say already, this is Nassau County, Suffolk County, North Shore, South Shore, East End. Brown tide. Well, a lot of Long Island doesn't experience brown tide. We only get it on the South Shore. And so, again, many of the sites that are problematic, these are the average levels over the last 2 years, are right by us on the South Shore. Quantuck Bay, Moneybog Bay, Penniman Cove, and other regions on the South Shore. And then fecal coliform, similar sorts of locations with some other areas popping up from the east end and the North Shore. And then the last of the parameters that we're studying is dissolved oxygen. I'm going to spend more time on that. You can see, again, some of the usual suspects showing up here, but I want to focus in on the absolute levels. The New York DEC wants to see, for the best health of our water bodies, more than 5 milligrams per liter of oxygen. And so what you can see is these are average levels. And so they want to see them never go below 5. And you can see many of these areas are having trouble keeping their average above 5. And so to hone in on this, I talked about epiphanies early on. We had another scientific epiphany in the last few years, something we've always known about, but that we're really recognized as giving us a different view of our bays, is that during the day, with the lights on, of course, algae and plants are photosynthesizing. And at night, that stops. So when there's no photosynthesis, there's no oxygen being put into the system, and instead, oxygen is being consumed. And so we've begun to, well, okay, I guess I'm not going to go to the, I mentioned 5 milligrams per liter. You'll see the standard here. This is from the New York State DEC. They're very serious for all their water bodies. They'll let it go below 5, but they don't want it less than 3 milligrams per liter. And there's good reason. There's science behind that, because when the levels go from 5 to below 3, you start to see mortality in larval fish and larval shellfish. And so they want it to always be above 3 and ideally above 5. So back to the epiphany, we've been putting out devices that are measuring oxygen all the time. And what they're showing is that the levels aren't static, but they change dramatically. And you see these lines. That's not bad data, that's real data. And that's what happens. During the day, the levels get very high. At night, they sag down. And so that happens every single day. Now, for 40 years, we've had very good monitoring data all across Long Island. People go out and measure oxygen levels, and we've had the impression we're in good shape. The problem is that those very hardworking people are working during the day when the oxygen levels are quite high. And it's at night when no one's out looking, but thankfully these devices are collecting data, the levels get so low. And so last year, anything you see across the line here in red and black is below that 3 milligrams per liter. The black is really below it. In 2015, anything you see here that's red, these are dates, June, going to the end of August. Here are 20-some-odd sites. All you need to see is that anything in the yellow is below anything in the orange or the red means that that is a point in time where we were not above the 5, or below the 3. So violating that standard. So it's widespread. Now, when I first showed this data to people, people said, well, okay, but you really want to verify that. We trust you, but if you want this data to hold up, you better get other people to work with you on it. And so I said, okay, that's a good idea. So last year we worked with the USGS. They have a nationwide monitoring program looking at places. They have federal standards that they match up to with regards to their measuring oxygen in water bodies. So we came up with very good quality control, quality assurance plan that started in September. Now, of course, the worst oxygen is when it's warmest. It starts getting warmer there. So you can see, if you go some locations, say Fire Island Inlet, oxygen here is chronically good. Right? This is all above 6. In this particular figure, anywhere you see 3, that's a day and point in time where it's below 3. Again, technically not allowable according to DEC rules. They never want to see that. And what you see is that this is Hempstead Harbor, Nassau County. You can see almost every day, this is time going here, this is time in hours, this is date going here. Almost every day below that level. And just a couple more sites, Northport on the North Shore, Hewlett Bay on the South Shore. So these oxygen levels are real, certified, verified, and a real problem when it comes to those standards, because clearly we're not meeting the standards in most of our water bodies, except for the areas that are very well flushed, like I showed you the Fire Island Inlet. So we need to think about what this means. We know what it means, because this is based on science. So now we need to think about how we address an issue like that. Okay. Hopefully we're more than halfway through. But everybody take a quick deep breath. Inhale and exhale. And so as you did that, you were taking in oxygen, and you were breathing out carbon dioxide, right? And we all know that bacteria respire just like you do. Bacteria do the same thing, and they're mainly responsible for the low DO levels. And everyone's been very focused on these low levels of oxygen. What we're recognizing and thinking about a lot now is the fact that the exhale from the bacteria and the input of CO2 is also very important, because that can affect pH. And so Ryan Wallace is here. Ryan, put your hand up. This is Ryan's groundbreaking paper. It's only been out for a little over a year. It's already been cited at least 30 times in international peer-reviewed literature. It's a very important finding, because we're really honed in on the idea that nutrients are not just leading to algal blooms leading to low DO, but they're also leading to high pH, the exhale, and they're also driving down the pH and acidifying our coastal waters. So that's why you see the name of the paper, Coastal Ocean Acidification. So it's the other end of what we call the eutrophication problem. And so Ryan, who had the nice laptop out there with the looping video, also has a poster out there, put together this beautiful view of Long Island Sound. This is New York City with depth, so 0 to 40 meters, looking at oxygen, pH, PCO2. We won't worry about what that is right now. And then this goes out to, I think, about Port Jefferson. But what you see, and what we discovered in this first paper, is that this low oxygen area in Long Island Sound also has very low pH, very high levels of CO2. In fact, for those of you who follow climate change, these are levels of CO2 that we don't even expect to occur in the open ocean even in 200 years. We then look back at data to look at the time course between oxygen and pH, and we saw that they're coupled. So the onset of low oxygen is in the summer, the western part of Long Island Sound, and that the pH follows a similar pattern, except that that low pH is more widespread, and it also hangs on longer into the fall. We've seen this time and again. And so it's not just Long Island Sound, but many eutrophic regions. Here's the Forge River on the south shore. This is pH versus oxygen, and you can see the relationship with the red dots. The lower the oxygen gets, the lower the pH gets. Now for this study, Andrew's here. He performed these experiments, Andrew Griffith. He added sodium carbonate to the water just to fix the pH. It didn't change the DO. It only made the pH higher. And because the question is, do we care? So our waters are acidified. Is that important at all? Because right now we're not thinking about that. We're just focused on the oxygen. And so in this experiment, this shows what happens when you just fix the pH only for bay scallop larvae. You can increase their survival by 4-fold. Or you just fix the pH for larval fish, and you can increase their survival by 4 to 5-fold. So the pH is important. And what this says then is that the pH in the Forge River is an impairment for survival of marine life. And just to show a few more highlights, these are hard clams, a little bit older, a little bit more hardy. If you give them low oxygen or low pH alone, they do okay. But both together is a problem. And remember, in the ecosystem, they're getting both together. And here's larval fish, a study published last year. And what this shows is that under great conditions, we can get about 75% survival. But if we mimic the pH and DO in an estuary, you can see the survival gets lower and lower. And when you mimic the actual condition, the fact that in the ecosystem they're getting low pH and low DO, you can see their survival gets extremely low. And so that pH is impairing fisheries, and I'm not going to say too much about that except to say that we need to now start thinking about pH for regulating our coastal waters. If we really want to protect our fisheries, we need to do better on oxygen, but we also now need to start thinking about pH. Okay, so I now want to shift focus a little bit and talk about victories. There's a lot of talk about improving water quality, and the question is that worth our time and effort? It's going to cost us a lot of money. Is it going to do anything? So I spent some time recently looking back at some recent data to try to get an answer to that. Try to go through this kind of quickly. But the first thing I'll just mention, if you haven't seen it, just last year, Suffolk County released its Comprehensive Water Resources Management Plan. And here's Steve Ballone, who has owned this issue of water quality. And so if you read the first 2 sentences of this report, the first one is wonderful. Water is the single most significant resource for which Suffolk County bears responsibility. That's owning the issue. And then also owning the fact that there are alarming trends in the quality of the water our families drink, as well as many water bodies. So this in and of itself is a victory. We've got a county executive who recognizes what's going on and is owning the issue. And to mention a few other things, before I talk about specific locations, people are now recognizing that there are water quality impairments. Statistical changes, according to surveying done by the Nature Conservancy, and I saw we have Marilyn here somewhere for the Nature Conservancy. There she is. Nature Conservancy doing great work on many fronts, but specifically looking at what the public perceptions are. They understand, public better understands these issues today. Our governor, our county executive, and I see Jay Schneiderman is here, as long as our former supervisor, Anna Throne-Holst. And so both of them, as well as our governor and our county executive, again, are owning this issue and becoming champions of this issue. We've gotten millions of dollars in federal funds to address this issue here on Long Island with regards to sewage treatment plants. Suffolk County is accelerating its program for installing alternative septic systems. I'm going to talk more about the Center for Clean Water Technology later, as well as the Long Island Nitrogen Action Plan. But to talk about some specific victories, we all know how important seagrass is. And this is from the Shinnecock Bay Restoration Program. I'll talk more about it. Christine Santora is here who helped collect this data. This shows where are the fish in Shinnecock Bay. The units are catch per unit effort. If you want to find fish, where are they? Well, there's your answer. So I could tell you that seagrass is important for fish, or you can just look at the data, and you can see that's where the fish are. We need our seagrass. Unfortunately, there's been an incredible loss of seagrass on Long Island since the 20th century. You can see here, we've already lost 90% of the seagrass that we once had. Okay? And that seagrass is valued at $1,000 an acre when it comes to yields of fisheries. So you can do the math on how much money we've lost through the years. Some people at the county think we'll lose all of it if we don't take action soon. And again, we know this is from excessive nitrogen loading. So where is the remaining seagrass? Well, the most lush seagrass beds are here in western Suffolk County and going into Nassau counties. That's western Great South Bay, South Oyster Bay. Here's the Bergen Point Sewage Treatment Plant, right? That's right there in that area. Now, that plant used to discharge directly into the bay. Around 1980, it started discharging out into the ocean. Did it matter? Well, here are the levels of inorganic nitrogen and how they've changed with time. And you can see this is before the ocean outfall. This is after. The levels have dropped significantly. Did that matter? The answer is yes. Here are surveys of seagrasses. The surveys in the green are areas that were present when the sewage treatment plant was discharging right into the bay. Okay? And the blue is what we have now. You move that strong nitrogen source, the seagrass comes back. Now, is this a fluke? I don't think so. This is what happened in that area. How about in areas where we didn't do anything about the nitrogen? And this is a gain of nearly 3,000 acres. Let's look at eastern Great South Bay. We didn't touch the nitrogen there. We just let it keep going. And we lost 4,000 acres of seagrass beds over the same exact period of time. So the bottom line is, if you fix the nitrogen problem, things like seagrass will respond in both ways. Let it go, you lose the seagrass. Take away the strong source, that seagrass can come back. And beyond the seagrass in that area, I'll also mention that there's one of the most robust oyster aquaculture facilities right in that same exact area. So the Blue Island oysters, they're doing very, very well. Aquaculture, for those of you who don't know, is on the rise here in Long Island, which I think is a wonderful thing. It's part of our heritage, and I think aquaculture is a way to bring it back as part of our heritage. And this facility is producing millions of oysters a year now, and those oysters are going all over Long Island, and specifically also in Manhattan and places like Brooklyn. And in an area that had sewage going directly into it. You couldn't harvest shellfish there, and now they're doing wonderfully. I also want to now hone in on Mumford Cove. This is an area in Connecticut, right? It's due north of Orient. small area that had sewage being discharged directly into it. And they also got it out, moved it to another location where things were better flushed. It's very almost identical story. The nitrogen levels go down to almost immeasurable during the summer, and here's the gain in seagrass with time. And so, to give a quick narrative, while the sewage was going into this particular area, the bottom was mainly macroalgae, ulva, sea lettuce. They had no seagrass, and they had closures due to that saxitoxin, PSP. It's one of the only areas that Connecticut was paying attention to. In 1987, they got the wastewater out. And slowly, within a year, the macroalgae covers begins to decline, seagrass begins to appear. They never had a PSP issue again. Once they got that out, they never had a closure. They test every year. I got the data. They're not finding any PSP. And then slowly the seagrass came back, 33%, and now half the cove is covered with seagrass. Very similar story. And how about Northport? This is on the North Shore of Long Island. This is an area that had been closed to PSP. You can see the years, 2006, 8, 9, 10, 11, 12, 13, over and over again. This is the worst spot on Long Island. They recognized the problem, and they addressed the sewage treatment plant. There was a sewage treatment plant discharging the back part of Northport Harbor in the exact area where they're having the most intense PSP problem, the most intense blooms. They got money to upgrade that plant. And you can see the work that was done to bring it down significantly. And here's the data. This is what the discharge was in kilograms of nitrogen per day. And here's the improvement of the plant. And you can see it just drops right off. And right now, they're discharging less than 5 kilograms of nitrogen per day. And concurrently, their PSP problem went away. Now, I can't say it's going to be gone forever, because we're still seeing Alexandrian there. But to show it another way, here are acres of shellfish beds closed in Northport and Huntington Bay, and here are the nitrogen levels. And you can see, once it drops off, the areas that are being closed, well, there were no areas closed. So I am under no illusions. There could be a bloom there again. This could come back up, but I don't think this is a fluke, based on what we've seen here and across the Sound in Mumford Cove. And beyond that, one of the worst bathing beaches that had been closed for 7 years reopened once they upgraded the sewage treatment plant. So great work by many great politicians in western Suffolk County, Doc Spencer and Frank Petrone. Doc Spencer, Suffolk County legislator, Frank Petrone, the supervisor of the town of Huntington, did a lot of work to get money to make this happen. So again, another success story. And then what about Long Island Sound? There's the biggest story of them all. Here's a plan that was put together in the 90s, reduced nitrogen levels by 60%. That's a big lift, a lot of money, and it took a lot of time, 20-year plan. And so what happened upon doing that? Well, let's look at the oxygen levels. Here's what the oxygen levels over the last 13 years, the hypoxic zone, the dead zone in Long Island Sound, going all the way back to the beginning of, turn of the century. And so here's what it looks like. There's some wiggle, but overall, the trend is getting lower and lower and lower. And I'll note, in this last year, in 2015, they've been monitoring going back to the 80s. This is the first time ever there was no dead zone in 2015. I should say this is at 2 milligrams per liter. If you said 3 milligrams per liter, yes, there was some below that. But it never got below 2 milligrams per liter. So again, people putting in a significant effort and the system responding. And coincidence? I don't know. But for the first time in decades, there were humpback whales in Long Island Sound last September. Not one, but multiple humpback whales. And in the spring, beluga whales. So, and again, these were the first time in a very long time. So these are hopeful signs that when effort is put in, the system will respond, it can respond, and recovery is possible. And I'll just go past that. And the final part of my talk is solutions. So we know there are problems. We know there are good examples. When people take action, there can be improvement. What should that improvement be? What should be done? So we know that septic tanks and cesspools are the largest source of nitrogen to Suffolk County coastal waters. And we know we've got 360,000 homes across Suffolk County that need to be addressed. That's It's a big lift. That's many, many locations. There's a Southwest Sewer District. That's one of the only regions that don't have very dense clustering of these systems. So I want to highlight some work, and I see Associate Director of the Center for Clean Water Technology, Jennifer Garvey, is here with us, doing a great job at the Center for Clean Water Technology at Stony Brook University. So I want to give you some insight into some of the advances that that center has been making in the last year. And so for those of you who don't know, one of the primary focuses of the center is to come up with alternative septic systems that do a better job of removing nitrogen, are more affordable, are simple to maintain, and hopefully have a small infrastructure. And it's supported by the DEC and also the Bloomberg Foundation. And so I mentioned this earlier on, our septic systems are very good at converting our wastewater into nitrate that goes to our groundwater and stays there. It's very, very stable on decadal timescales. However, and here's our typical system that we have here on Long Island. Most people have septic tanks that then go to leaching pools. Those leaching pools leaching their contents, hopefully not into your drinking water, but that can happen. And I live in an area where I've got my own well, and so I need to worry about that. And there are the greatest cluster of those sorts of homes are here on the East End. But not all systems need to be like that. There's this wonderful process of denitrification that can convert this nitrogen into nitrogen gas that goes to the atmosphere. 80% of what we're breathing is nitrogen gas, totally innocuous. And so we want to have systems that do this. Don't let the nitrogen go to groundwater, but rather get it out of the groundwater. And so right now, Suffolk County is piloting a series of systems that do this. They're being piloted 6 different systems in 20 locations, and they plan on making these approvable for homeowners in this calendar year. I've heard a couple different dates, June, December. I'll go with December, based on the way things move, but we'll see. I want to introduce to you, however, some alternative systems that I think are on the horizon, hopefully, for Suffolk County. And the first system is something known as a drain field, where instead of the waste going from a septic tank into a deep leaching pit, it goes into a series of tubes here, PVC pipe that's perforated, that slowly leaches through into the soil. Now, interestingly, these are not permitted on Long Island. And we had some scientists from across the country out at Stony Brook earlier today, and had a conversation with one of them, and he just remarked how astonished he was that those don't exist in Suffolk County. Everywhere else in the country, maybe in the world, if you don't have a sewage treatment plant, at least you'll have a drain field and not one of these deep leaching pits that lets the nitrogen go right to the bottom. The deeper you get in our aquifer, the less nitrogen is removed. If these pipes are up here, your lawn can take up some of it, trees can take up some of it, and the temperatures during 6 to 8 months of the year are warm enough that you have enhanced microbial processing. Bacteria can chew up some of that nitrogen as well. And as sea level rises, we need systems like this because our deep leaching pits are going to get inundated with groundwater and with seawater. What we're working on the Center for Clean Water Technology is the next generation of these drain fields. It's pretty simple. It's the idea that the pipes from the drain fields go through a layer of sand and then go through a layer of sand mixed with sawdust. So here's the aerial view. I'm going to now show you a side view. So there's the pipe, influent coming in, layer of sand mixed with some sort of either sawdust or wood chips. So the interesting thing is the sand takes care of one nitrogen reaction, makes nitrate. When you have the sawdust in there, it converts that nitrate into that nitrogen gas. And so this seems so simple, right? How simple could it be? But it is so effective. Well, I'll just show you this. Part of the punchline here. I'll show you some data in a moment, but it gets down to 3 milligrams per liter. That's what a sewage treatment plant does. Engineers call that the limit of technology, and that's based on just having a pipe and some sand and wood in the ground. What does it look like? Here's one of them being dug out. Here's one of them completed. And then here's a drain field with the grass beginning to grow. If you don't have something else to disperse that waste, you can have striping, or you can have a really green lawn. Really green lawn. Fertilizer not needed. here's some data, and there's more data of this, but here's the nitrogen levels going into the system. Here it is coming out. And again, on average, and this the data keeps going and stays right here, on average 4 milligrams per liter is the long-term average on this system. And again, as good as a sewage treatment plant. And here's some data. I see Stuart. Well, I keep wanting to say his name. There's Stuart. Here's Stuart's data. He's a postdoctoral scholar at Stony Brook University, generated this data. 2 different systems in Massachusetts. You can just look to the punchline here. 80 to 90% removal of nitrogen in these systems during the dead of winter. How cold was it, Stuart? 5 centigrade or Fahrenheit?
It was almost 0.
Yeah. And so these systems would be performing at their worst because it's cold. I told you the microbes are temperature dependent. But even at that temperature, they're removing most of the nitrogen. Just talk about some other systems that we'll be working on as well. Something known as constructed wetlands that can also do a good job of removing nitrogen. And something known as permeably reactive barriers. This is a situation whereby at the cat's out of the bag, so to speak, and you already have a plume of nitrate. A barrier can be put in along a coastal ecosystem to intercept the flow of groundwater and essentially do the same thing. You put that sawdust in here in the vertical, and it will do the same sort of thing, remove that nitrogen before it gets out into the ecosystem. And pilot studies have shown up to 90% removal up in Massachusetts. And so some things to think about on the wastewater front as we move forward. Firstly, there are already small sewage treatment plants that can be put into place now for treating nitrogen. That's for large volume flow. And again, by the end of 2016, these individual systems will be available, not the ones we're working on at Stony Brook, but the commercially available ones. These drain fields only make sense for Long Island, and I think need to be part of our future toolbox for dealing with wastewater. But the big thing going forward will be funding. Any sort of installations will cost a lot. I should say, those drain fields, hopefully you got the sense, that's pretty simple technology. That's a fairly simple install, not as costly. Some of these other alternative systems that people first began talking about, people would throw around the number $50,000 per home, right? Who's the homeowner? Raise your hand if you're the homeowner who wants to pay that bill. Yeah. But these other ones are more similar to the price you'd pay to install a standard system, maybe a few $1,000s more. But so this shows promise not only being efficient, but also being significantly more cost effective. I told you another of there are plans currently to reduce nitrogen levels in Long Island, and I just want to, so there's the Long Island Nitrogen Management Plan that the DEC has kicked off, and one of the issues to address there is where should the nitrogen removal begin and how much should we reduce it. And so I just want to talk about a study that I'm engaged in collaboratively with the DEC, Suffolk County, and the USGS. Ryan Anderson is not here, but he's also engaged in this study. And the idea is to figure out all of the watersheds with USGS across all of Long Island. There's going to be hundreds that we consider. For each one of those, come up with the exact nitrogen loading rate. Also, for each one of them, figure out the receiving water body. What's the residence time? How's that water moving? I'll talk about that momentarily. And then together come up with something we call a nitrogen residence time. Once the nitrogen hits there, is it staying in the bay for a long time, festering and manifesting itself in all these negative ways, or is it being flushed out right away? Which will then facilitate a ranking of watersheds that are most in need of being addressed first. And then concurrently, we can just look at the standing data of water quality, which should give us a sense of what should be addressed. Should line up perfectly with this. And Suffolk County would like to see the first 50,000 homes to be addressed, to be identified through the study. Wrapping up, I'm going to just talk about, I think, just a couple more things very quickly. This is a matrix from Cape Cod, where they've been dealing with the same thing we have. And they just went through a very intensive study period where they came up with a couple of different conclusions. One, there's many different ways to deal with nitrogen loads. And two, when you address the wastewater issue on land, that's great, but it's going to take, in some cases, decades for that effect to be seen in the water, our coastal waters, because it can take decades for water very far inland to make its way to our coastal water bodies. So in the meanwhile, we need to address the issues that are happening in our water right now. And so I'm going to talk about a couple of those very quickly. One that we're thinking about here is what role can seaweeds play in removing nitrogen? Where's Craig Young? There he is. Raise your hand, Craig. He's done a great job in this study, essentially looking at how macroalgae can take up nitrogen and phosphorus. So in many parts of the world, aquaculture is a staple of harvesting from the sea, be it for finfish, shellfish, also for macroalgae. And for those of you who don't know, here's the comparison of wild capture fisheries and aquaculture over from the middle of the 20th century onward. And the bottom line is we've now entered the era where most of the food that we eat from the ocean is not wild captured, but is aquacultured. And that trend's just going to continue. We don't do a great job of that here in the U.S., but we could. Could do a better job, and I think, like I said, I think there's a trend in Long Island to see more and more of that. And so Craig's been working with a couple of different species of macroalgae. He's been growing this out in Shinnecock Bay over the past several years, starting in May, going all the way into the fall. And so in growing it, he's been able to estimate how much nitrogen, if you put these different algae out week by week and harvest them, he's figured out they replace themselves in a week. They double in growth. They grow very, very quickly. And so after a week of harvesting harvesting week by week through the summer and going into fall, you can remove a significant amount of nitrogen if you have multiple ropes of these different macroalgal species out, and we're going to be expanding that this summer. In addition, work through the Shinnecock Bay Restoration Program has been out trawling, looking at fish populations, and incidentally pulls up lots of macroalgae, seaweeds that are already out in the bay. And so here's a map. We now know through all these trawls where we're finding these macroalgae, and we found a few spots that consistently have very high levels of macroalgae. Now, if left to stay there, these macroalgae that have taken up lots of nitrogen and phosphorus will just decay away, release the nitrogen and phosphorus right back to the bay. Alternatively, this can be extracted from the bay and pulled out, and if doing so, you're removing a significant nitrogen source. So again, Craig did some trawling last year and looked at if you were to target particular regions and pull out the amount of the macroalgae on a regular basis, going from summer into fall, how much nitrogen could you remove? And so this is expressed here as a percentage of the fertilizer nitrogen that goes into Shinnecock Bay. Okay, so what you see is that in a particular area in Shinnecock Hills where we're seeing a lot of macroalgae, the consistent harvest of that macroalgae could take out more than 50%, more than half of our fertilizer-based nitrogen. But wait, it gets better. What if you remove that and turn that into fertilizer? Then that would take care of the other half that's being used. And in fact, this gets done in other parts of the world, in Europe. And this would be an organic, renewable, non-polluting resource. So instead of the nitrogen from our fertilizer wreaking havoc and leading to destroying our fisheries, you could make that a renewable resource. And that's something we're going to be working on more in the coming year. Jay Schneiderman is here, our supervisor, and is very interested in ocean flushing for good reason. And so I think almost the last thing I'm going to show here are the occurrence of brown tide in Quantuck Bay over the last 30 years. Year after year, this place is having brown tide blooms. And very quickly, I'm just going to show you, we now know, in addition to nitrogen loading, why that is. This is a movie releasing a tracer into Quantuck Bay. We built what's known as a hydrodynamic model. And it is a model that describes the flushing of Quantuck Bay, Moriches Bay, and Shinnecock Bay. The data is based on the bottom topography, the depth of the water, the known salinity of the water, which gives you a sense of the mixing with the ocean, tidal information that's available from NOAA. And so what you're looking at now, that tracer was released 1 week. If you're following, these are the times here. So now we're almost getting towards 8 days. And so 8 days after the water's released there, it's not even close to the Shinnecock Inlet, and it also hasn't made it to the Moriches Inlet. I'm not going to let this thing go all the way through. It goes for a month. But the bottom line is, to get out of Shinnecock Bay, it takes almost 3-4 weeks, up to a month. So the bottom line is that this area is incredibly stagnant. And remember the News 12 data I showed you, and that porous water quality keeps coming up in this particular region. And so what happens when you have better ocean flushing? Well, I'm not going to show a lot of data, but here's a beautiful picture of the new inlet and Great South Bay. This is what the rest of the bay looks like. By the inlet, it's being very well flushed. And similarly, here's another aerial photo. I'm not sure whether you can see it, but this is brown tide being flushed out to the ocean by this ocean inlet. And here's some data all across the South Shore during a brown tide. This is 2013. But just to show that these ocean inlets keep the brown tide at bay. So ocean flushing is very effective. A final data set to show on this. Here's East Hampton, Georgica Pond, where we've been working. You may know this is open to the ocean a few times a year, specifically in the fall. And here's what happened last year when they opened it to the ocean. Very serious blue-green algae blooms, some of the highest densities we saw in all of New York State. With the inlet opening, it gets flushed right out. And we saw that in 2014 as well. Coloration here, red is more intense, purple is the least intense. You can see the two dates, opening that inlet, flush to the blue-green algae right out of the pond. The very last thing I'm going to show is the work for the Shinnecock Bay restoration programs on bivalves. I see that we've got Ed Warner and Scott Horowitz here, the Southampton Town trustees, who have been a wonderful partner in this project. Through their efforts, they created the two largest hard clam spawner sanctuaries on the eastern end of Long Island. So these are no-take zones where we can put clams in there to grow, reproduce, and make more clams. That's been incredibly important, and I'm not going to show you the data, but I'll tell you it's been highly successful. The clams are doing very well there. We've also spun up our activities at our new marine lab. For those of you who haven't seen it, across the street, we had a $10 million marine lab built just a couple of years ago. Significantly expanded our ability to culture shellfish. Andrew Griffith is here, who's done a wonderful job at pioneering our work and making these guys reproduce. So here is a clam caught in the act of reproducing. Is this a male or a female, Andrew? Female. Female, okay. So we get them to spawn individually. We then mix the males and females to create these larvae. So those of you who don't know, this is what a clam looks like in its first weeks of life. And so Andrew and others in the lab, I see Brooke, now Alexandra as well, have done a wonderful job working on these bivalves, getting them to spawn and reproduce. And we've been making bags of shell, again, thanks to the Southampton Town trustees who have collected the shell for us to use. These are just simple bags of shell that we then set oyster larvae on. So this is a shell, and here's a day. Andrew takes those larvae that he grows out, puts them on the shell. You can see them here. Here's one day after they set on that shell. They're chemotaxic. They seek out the shell. They want to grow on the shell. Here's a week after, and here's a month after. So you can have one shell with literally 50 oysters growing on it. This is how they grow naturally, like you hear about oyster reefs. And that's what these are destined for. In fact, we have the first, DEC tells me that they're in the process of approving our permits for the first oyster reefs. I don't believe, well, the first one's on Long Island. They told me in New York State. I said, well, what about the ones in the harbor? And I don't remember what his answer was, but we'll say on Long Island. So our permits are being, in the process of being approved to make the first oyster reefs. They'll be in Shinnecock Bay, I'm hopeful, by the end of this year. And that's what these shell bags are destined for, because that's what they're growing as, on shell. And so through this program, we've produced, and again, in collaboration with the Southampton Town trustees, more than 7 million shellfish in Shinnecock Bay, and we're extremely excited to see last fall people just haphazardly sampling Shinnecock Bay, finding lots of juvenile clams. Some of the species that we're targeting, some of other species, but it's a good sign. And that's what these spawner sanctuaries and activities are designed to do. So I'm done. I'll conclude by saying we know excessive nitrogen loading is an ongoing threat to coastal ecosystems, our economies, pets, and human health. Nitrogen reductions, I've shown you, are leading to significant improvements in ecological conditions in many of our coastal water bodies where we make the effort. When we don't make the effort, it goes in the other direction. And so we now need a coordinated and multifaceted efforts both on land and in the water to restore Long Island's coastal waters. So with that, I'll thank you very much for your attention. Thank you. And I'd be happy, let's have some questions, and I'd be happy to answer those questions. Bob. Yes.
What is any idea of the life of the system?
So the question is, how long do those drain field systems last? And I see my colleagues giggling because we've been in a meeting the last 2 days with scientists from around the world trying to answer that precise question. And so theoretically, we think they could last 50 years or more. There's some nuance to that, and there's some unknowns, but for some particular systems, my colleagues would want me to say, for lined and saturated systems, the wood should last a very long time because the oxygen levels are zero and there's low degradation when there's low oxygen. Yes, Joan.
How much space does one of those systems require?
So the question is, how much space do they require? And the answer is, it depends. They can be, the systems that we've been working on, some of those systems, I should have said, we've been working on some of those systems at this test facility in Massachusetts. And they're generally about 20 by 20. They can be made in different sizes. They can be made smaller if you feed the wastewater at a more rapid rate, or you make them deeper. But right now they're being piloted about 20 by 20. And, as you can see, effective. Yes?
Chris, why have drain fields not been used?
The question is, why have drain fields not been used? And so this is sort of a mystery, but not entirely. The health department is responsible for wastewater in Suffolk County. And so their primary concern was to make sure that any pathogens associated with wastewater get as far away from the citizens of Suffolk County as possible. So they sent it down very, very deep. And so they've done a very good job of making sure that people aren't getting sick. But in hindsight, those same systems have led to that rising level of nitrogen in our groundwater. But that's the best answer, and looking back as to why that is. Yes?
It looked like those permeable reactive barriers have to go down into the groundwater.
That is correct.
How far, and if it's made out of sawdust and sand, why doesn't the sawdust just float up to the top?
So when these are made, the area's got to be, those permeably reactive, the question is about permeably reactive barriers and how well they function and how stable they are as they're being put in. A trench needs to be built, and there needs to be a sort of a dike or a dam to hold back the groundwater as they're put in. I can't remember the first part of your question now. How deep? So the how deep depends on the nitrogen that you want to treat. So theoretically, I'm working on a project on this in East Hampton, for example. And so the first thing we're doing is we're trying to map out where the nitrate plume is in 2D. So we want to know both the extent horizontally and also the depth vertically. So you can make it any size you want. Of course, the larger it is, the more effort you need to be putting in. But it's primarily designed to hit surface groundwater, and if put at the coastline, groundwater, even that's deeper, at least in the upper glacial aquifer, rises up towards the surface. So technically, you wouldn't need to go more than 10 to 20 feet. And the technology for putting these things, there are companies now that have some innovative ways to make sure that the groundwater doesn't inflow that quickly, and these things can be installed properly. Yes, Charlie.
Well, my question is twofold, actually. The walls that you were just talking about, do they absorb the nitrogen?
They don't. So I keep mentioning wood, sawdust, wood chips. The idea is that, and that's the same that goes in the permeable reactive barrier, that wood encourages bacteria to undergo denitrification. They use the carbon source, the oxygen goes to zero, and that's the conditions you want. And also, by having the right permeability, the right sort of, say, sand in there, as well as the wood chips, it actually can wick water in. So if developed properly, the water will seek the path of least resistance and actually purposely go through that.
See, that's exactly what my question is, though, because I get that the pipe system kind of works the same way that a seagrass bed would, or a salt marsh, I should say, where, denitrification happens there because it gets flooded, because the rhizomes at the bottom of the plants that live there, there's one always underwater, how are these bacteria taking what's not nitrate and turning it into nitrogen gas?
It is nitrate. That's the whole thing. Yeah. Long Island groundwater, once you get away from the septic tank, is fully oxygenated and fully turned into nitrate. Yeah.
So is,
Let's give somebody else a chance. We can talk further afterwards. So, yes, Ron.
Are there methods being considered for flushing the bays as the short-term solution to the current nitrogen?
There are things being considered. I know that, like I said, the supervisor is interested in that and is working with John Bouvier, town board member, exploring options for doing that in areas where it's needed. It's complex because you're dealing with the energy of the ocean on the one side, but there is some precedent for that. So it's being considered by wise people. Yes.
Thank you, Dr. Gilbert. Speaking of coordinated multifaceted efforts, I think it's a good idea to coordinate the felling of fish plants to combat the spread of southern pipe beetle. All this sawdust we're going to need next year to facilitate this.
Well, it's funny you said that because we met, part of the people in our meeting today was Carrie Gallagher, the Long Island head of the DEC, and that actually came up in conversation. And so and something we're very seriously considering, because what we want to do is, for these drain fields, is make sure we're using all locally sourced material. So it only makes sense, if we're serious about this, that we should be testing Long Island Sound and Long Island sands, and Long Island woods, so pine, oak, and maples. So the answer is yes. Yes.
Sorry to hit you again, but East Hampton Town just noticed for hearing a draft law requiring intermediate-sized systems in the Harbor Protection Overlay District to upgrade their systems within 5 years, down to 3 parts per million of nitrate coming out of the leaching pool. As owner of 2 of those places, and what we've been talking over the last week, the hearing, by the way, is this coming Friday. So
Noted.
It's rather urgent and on topic right now. So we've been talking to vendors, and they've said anything from $50,000 to $100,000 to implement an intermediate-sized system, and that the operating cost could be over $50,000 a year because a Suffolk County licensed engineer has to come on a daily basis to check it.
That last part's not correct. Yeah, not that frequently for sure.
Your perspective on the bureaucratic potential barriers to implementing something like that with the county and the townships, and also, the proposed legislation in East Hampton doesn't seem to allow for creative net zero solutions, like putting ropes in the water and harvesting algae, or having waterless urinals that segregate urea, which would take, I think urine's 90% of the nitrogen. So
Close to it, yeah.
if you take half the population,
Yeah.
and you just put that into a holding tank, and then you use it for fertilizer on top,
Yeah.
that's 45% reduction
So-
with very little cost.
Yeah, when Cape Cod did their 208 study, I showed you that matrix, they did come up with multiple solutions. And so I think, that every town and all the county, this is all very new. And so everyone's finding their way. I shocked myself when I went back in my notes and saw that when Steve Ballone, our county executive, for the first time took ownership of the nitrogen issue, it was 2014. I thought it was, a long time ago. And so this is all very new. And so I think it sounds like the situation in East Hampton is one that will probably play out, where I think everyone's going to have to think thoughtfully and carefully about the best way forward. I will just say, with regard to your individual system, there's probably opportunities for getting multiple people, or I don't know exactly where yours would be located, but there's no reason for us, in my mind, knowing the capacity of those systems and guessing the flow out of your facility, that you would have to be the sole person responsible. There's probably opportunities for the town to get other people hooked up, And there needs to be, the most important thing, there's going to need to be some significant financing for these things that can't be burdened by just individuals, just one business. But again, everyone's feeling their way. So I think I have no doubt there's going to be fits and starts, and it's going to take creative solutions. I don't want people to be sitting for too long. I'll just take a couple more questions. Yes?
Thank you so much. This was a really heartening conversation, and the facts are really encouraging. My concern remains that having these new systems will give excuse for increased development. And the problem is that not only will that shift into more waste, but other kinds of pollution like noise and air pollution.
And you're not alone in that concern. Many people have that concern. And so I think some of the answers are that it doesn't make any sense to have, there's going to be the total nitrogen loading study. And so we're going to now know exactly all the nitrogen going in. And so if there's improvements, we'll know what that is. But if there's more homes coming in, we'll know what that is as well. And so it won't make sense to reduce nitrogen only to let it go back up. That's the one thing. The other thing that's very important is that a lot of regions are, the ability to develop is based on zoning. And so, I think that needs to probably expand even further. And that is to say, this is an area that is 2-acre zoning, 5-acre zoning, 1-acre zoning. And when that sort of situation is put together, it doesn't matter how the wastewater is handled. It's based on the zoning. And so I think in many areas, I know in the town of Southampton, people have put in a significant effort to make sure that areas are zoned. And when an area is zoned, it is what it is. And even if you improve wastewater, that won't let people. The zoning is what trumps is the trump card there.
Except you forget the PDDs.
I won't discuss PDDs. I'll take one last question.
Long Island is the most crowded large island in the world. Every place else with higher population density is either a really big city or a tiny speck of a fishing village that's productive or something. Aside from the fact that, as was alluded a question or two ago, we're not really going to have, we're certainly not, we're far more likely to have more people on Long Island, the whole of Long Island, urban, suburban, and rural. And less, but in terms of the kind of policy that you like to see, and that's a pleasant surprise to see someone at the level of a county executive actually do take ownership of water, which is kind of like the most essential and least talked about feature of what a government is supposed to do, although we now know that government doesn't necessarily do that the right way or at all for instance. However, as Long Island and its surrounding waters, the Sound and the smaller groundwater areas, it has had a reputation, for better and worse, of being ahead of the curve, or the canary in the coal mine for people doing coastal estuary back bay waters in North Carolina or Texas or Washington State, because what is going to happen to them over the next 50 years already happened here 50 or 100 years ago. I guess I'm just curious, in terms of public policy, are any of those places doing what you would like to do, or is this, or is this place and this research still the bleeding edge around the country and beyond?
Good question. And I think there are what's been exciting about doing this research, and specifically at the Center for Clean Water Technology, is that we've begun to make connections with other parts of the United States. So some of the scientists we were working with today were from Rhode Island, from Massachusetts, from Florida, and they're dealing with the same issue.